Hello and welcome, everyone. My name is Jessica Blaustein. I'm the Senior Director of the Healthcare Partnerships team at the Muscular Dystrophy Association. Thank you for joining us today for this Industry Update webinar, and thank you for all that you do for MDA and the neuromuscular community. This webinar will share information about investigational RGX 202 and is titled Affinity Duchenne, a Pivotal Trial in Duchenne Muscular Dystrophy. This program is sponsored by REGENXBIO. MDA's Industry Update webinars provide an opportunity for the medical community to receive information and updates impacting care and treatment directly from pharmaceutical and biotechnology companies. These webinars are just a part of MDA's medical education resources. We encourage you to visit the medical education pages of mda.org. You'll find resources including MDA's Clinical Research Awareness Program, Grand Rounds webinars, our MDA monthly report e-newsletter, and much more. Now, I'd like to introduce our presenters for today's webinar. We'll hear from Steve Picola, MD, Chief Medical Officer at REGENXBIO, Naz Dastgir, DO, Senior Medical Director, Clinical Development of the DMD program at REGENXBIO, and Nancy Kuntz, MD, Lurie Children's Hospital, Professor of Pediatrics and Neurology, and Affinity Duchenne, PI. During the presentation, please submit any questions you may have into the Q&A. Time permitting, the team will address these at the end of the presentation. And now, without further ado, I'd like to turn it over to Dr. Picola.
Thank you, Jessica. My name is Steve Picola, Chief Medical Officer at REGENXBIO, and I first want to say thank you to MDA for this opportunity to provide an update on RGX 202, our investigational gene therapy for the treatment of Duchenne muscular dystrophy. This slide shows our objectives for the next 30 minutes, where we'll inform a bit on the program sponsor, REGENXBIO, and dive right into background and clinical data on RGX 202, and lastly, discuss the currently enrolling phase 3 pivotal trial. And we'll achieve this with this program, where after my introduction, I'll hand it over to Drs. Naz Dastgir and Dr. Nancy Kuntz to review the phase 1/2 trial design and results, and the design of the pivotal 202 trial, before we finish with Q&A. So first, who is REGENXBIO? We've actually been a leader in the gene therapy space for over a decade now, based on our own AAV vectors, which are the most clinically validated vectors for safe and effective gene therapy treatment for various indications. And notably, this includes the first ever approved systemically administered in vivo gene therapy, Zolgensma, for the treatment of SMA. This slide shows our internal pipeline with 3 late-stage programs, all in or through pivotal trials, including our lead program, RGX 202. Notably, we manufacture all of our products in-house in our state-of-the-art manufacturing facility, and we already are making commercial-ready product that is used in all our clinical trials, including for 202. We use the best-in-class suspension cell-based process and achieve very high purity with a greater than 80% full to empty capsid ratio. Now we move to the focus of this webinar, RGX 202, the investigational one-time gene therapy for the treatment of Duchenne. And to provide background on 202 and its unique differentiating characteristics, we have with us Dr. Naz Dastgir. Naz is the clinical development leader of the 202 program. She also is a practicing pediatric neuromuscular specialist, and this brings real-world perspective to the treatment of patients with Duchenne. Naz, take it away.
Thanks, Steve, and thank you again to the MDA for having us here today. So what we're going to go through a little bit is some of the preclinical data that supported developing our drug RGX 202 for the treatment of Duchenne muscular dystrophy. So preclinical refers to obviously our mouse studies that we did. And what we found with our unique transgene, which I'll go through a bit more details on the next slide on, is that with the addition of a CT domain, the C-terminal domain, we derived some additional functional benefit in the mouse. So we saw that the CT domain significantly enhanced restoration of the dystrophin-associated protein complex in the MDX mouse and made it more similar to natural dystrophin. It prolonged the microdystrophin activity. So the CT domain itself increased the half-life of RGX 202 microdystrophin, which remained in the muscle fibers longer to strengthen the muscle. And it also helped with muscle health. So the CT domain in RGX-202 microdystrophin protected against contraction-induced damage, enabling better muscle recovery. And to the right, you can see some of the mouse studies that we did. So on the y-axis here, you can see the percent initial force that was generated by the mice after they did 20 eccentric contractions. And in a control mouse with no DMD, you saw that there was some reduction in force, but to about 70%. But in the DMD mouse, it goes below 20%. And when we gave our drug without the CT domain, we hit around 40%. But with the CT domain, we were able to generate more force, closer to 60%. And RGX-202 is the only microdystrophin gene therapy currently available either research or commercially with the functional elements of the C-terminal domain, which is found in naturally occurring dystrophin. So here is a nice comparator slide of what our actual transgene is compared to other available transgenes that are out there, either commercially or from a research standpoint. So we do use AAV8, so it's AAV8 is our vector. Our promoter is SPC512, and obviously the transgene was developed to retain the key elements that we think would be essential for good preservation of the sarcolemma and better function. And this includes the unique features that you see here, and also the most unique feature, which is the C-terminal domain. And RGX-202 is an AAV gene therapy that is administered as a one-time dose. So once you receive it, there's no additional doses. The transgene is encapsulated within the AAV vector and injected as a one-time IV infusion to the child. So I'm going to go through a little bit of our clinical study. The title of the clinical study is known as AFFINITY DUCHENNE. And we'll go through some details of where we're at, and I'll pass it on to Dr. Kuntz for more details on some of the early readouts that we've had. So we had a phase 1/2 trial that is actually still enrolling in one portion of it, so I'll tell you more details about that. The key eligibility criteria was to enroll boys who were 1 to less than 12 years of age at screening. They needed to have genetically confirmed Duchenne muscular dystrophy that included mutations in exons 18 and above, and have no pre-existing antibodies to the AAV8 capsid. The inclusion criteria was a little different for the different age groups. For those children who were 1 to less than 4 years of age, they just needed to be able to complete the 10-meter walk without assistance, be on a stable dose on or off corticosteroids for 12 weeks, and have a weight greater than 10 kilograms. And that weight greater than 10 kilograms just comes from blood volume restrictions in terms of safety lab monitoring in the young children. And in the 4 and over children, they needed to be able to complete a 100-meter walk without assistance and be on a stable dose of corticosteroids for at least 12 weeks prior to entry into the study. We started off with a dose evaluation phase. So we first started with a dose of 1 × 10⁻⁴ genome copies per kilogram. We dosed 3 children with that dose and had an external monitoring committee that said, okay, good, safety data is good, let's go on to dose level 2. We dosed 2 children with dose level 2. Everything looked great from a safety standpoint. So we got the okay to go on to an expanded cohort where we enrolled an additional 5 patients. Those are all completed. But since we were doing well with the 4 and over patient cohort, we decided, you know what, especially with newborn screening that's now available, let's address another group that has pretty much an unmet need, which is the 1 to 3-year-old patients. And so we opened up a separate cohort for up to 5 patients that are now receiving 2 × 10⁻⁴ genome copies per kilogram. So we are still enrolling in that cohort at certain sites that are still running that version of the protocol. And so we have room to enroll more patients there, and that's why there's not a check mark there to say it's fully enrolled. So there's still some time in that cohort right there. And the trial design is listed on this timeline here. So in the light gray, you could see the pre-dosing section with RGX 202 administration being day 1. And then the post-dosing monitoring that happened in the phase 2 study was from day 1 to 12 months after dosing. That's in the darker gray. And then we go to a lighter gray for the long-term follow-up that's required by FDA for 5 years after dosing. So in the pre-dosing phase, what we do is get a whole bunch of baseline assessments, which include muscle biopsy, baseline muscle function tests, which are a bit different for the different age groups. So the key ones are listed here. So in the 4 and over age group, we have time function tests like time to stand, time to run, walk, time to climb, and the NSAA, the North Star Ambulatory Assessment. And then the 1 to less than 4-year-old age group, we use the Peabody Developmental Motor Scale Type 3. And the biopsy is to get the baseline biopsy on these patients. Obviously, these children are not going to have any microdystrophin at baseline, but we like to see what the fibers look like, what the baseline, if there's any dystrophin positivity, for example, and get the baseline for comparison to a follow-up muscle biopsy that occurs 3 months after dosing. At baseline, in children who are over the age of 4, who can tolerate these procedures without sedation, we do MRIs. So this would be skeletal and cardiac MRIs. And at that time, pre-dosing, we also initiate something called an immune suppression regimen. So you're probably familiar with this with regard to corticosteroids being initiated. This is steroids given on top of the baseline steroid regimen. But we do 2 additional drugs on top of that, and that includes sirolimus and eculizumab. This is finite dosing of drug. Eculizumab is administered, we give a couple weight-based administration schedule. So we give a couple doses before day 1, which is the RGX 202 administration. And then we give a few afterwards, and then you're done with the eculizumab. And we usually give full dose of the sirolimus and the additional prednisone or prednisolone for 2 months and then wean off in the 3rd month. And we find that that is a very well-tolerated regimen, and it has helped with our tremendous safety profile that we've had to date. And I'm going to pass this on to Dr. Kuntz to talk a little bit more about the baseline demographics of our patients and some of the early data that we've seen.
Thank you, Naz. I apologize, Chicago's a little noisy. I'm going to spend the next few moments describing the baseline characteristics of the cohorts that Naz has just described, and give you some of the data from the first or most recent data cut. So looking at this slide, you can see the baseline demographics outlined for both dose level 1 and dose level 2. For dose level 1, the average age at dosing was 7.1 years, with the youngest dose being 4.4 and the oldest 10.5 years of age. There were 3 children in this dosage level. The average weight of these 3 was 24.3 kilos. And you can see that the duration of follow-up reached a mean of 17 months. And then looking as they moved on to dose level 2, which was 2 times 10 to the 14th genome copies per kilo, you can see that there was a single child in this dose cut of under 4 years of age. And the exact characteristics of that child are listed here. That there were 7 subjects in the 4 to 11 age group. And for these, the average age at dosing was 8.7 years, with an average weight of 26.2 kilos. You'll note that the largest weight of these subjects was 35.5, and an average follow-up period from time of dosing of 7 months. So here are the demographics. Next. Okay. This slide demonstrates the safety that has been observed in these cohorts. And you can see that RGX 202 was very well tolerated in all of the patients treated to date, including those dosed with dose level 2. And that includes the single child under the age of 4 included in this cohort. In all of these cohorts, there have been no serious adverse events, no adverse events of special interest, and no cases or evidence of myocarditis or myositis. The most common drug-related adverse events were those reported commonly in any kind of gene therapy trials, and that included nausea, vomiting, and fatigue, all of which were self-limited and resolved. Okay, next slide. Here we go. Biomarkers were followed, and they supported both the transduction and expression of RGX 202 microdystrophin. And you can see these on this graph or figure separated out for dose level 1 and dose level 2. I'd like to highlight a few key points. At dose level 1, in the children ages 4 to 7, the average microdystrophin expression measured by Western blot was 60.6%. And you can see in the single child in this lower dose level, who was between 8 and 11, his microdystrophin level was 10.4%. Moving to the right at dose level 2, you can see that in the 4 to 7-year-old group, the microdystrophin level on that 3-month biopsy was 77.2%. And in the group that were between 8 and 11 years of age, and there were 5 in that group, the microdystrophin level was 39.7%. There were some additional features to support expression of the microdystrophin, and this was both assessment of the vector copy number and the percentage of positive fibers on immunofluorescence on the muscle biopsy. And you can see those numbers listed here. Next slide. Before we talk about the interim functional data on the data cut for the phase 1 and 2, I'd like to talk a little bit about the methodology used for the external natural history control. Again, all of the subjects in this trial were treated with a product, and the control comes from an external natural history control. You can see on the left a figure that I think most of us are very familiar with, which is the natural history or trajectory of motor function, here reflected by the NSAA score in boys with Duchenne muscular dystrophy, with the x-axis indicating the age. You can see first the slowing of progress, then the plateau, and then the decline in motor function. Okay. The method for external controls in this trial was a natural history control where age and then the baseline function using these timed motor tests, time to stand, 10-meter walk run, and time to climb 4 stairs. And those were matched very carefully with the individuals in the trial. So this is the control for the data that you'll see in the next slide. Here, we're just, this entire figure is dealing with dose level 1, okay? And here, the blue bars show the average mean change from baseline to the 12-month reassessment for the timed functional tests. And on the left, you see time to stand, 10-meter walk run, and time to climb the 4 stairs. And on the right is the NSAA scores as they changed. There were only 3 patients in this cohort at dose level 1, but you can see that the gray bars indicate what would be expected in a group from the natural history study. And so in the natural history study for all of these time function tests and the NSAA, there were losses. But in each of the, in all of the subjects, and you can see the mean scores here, there was an improvement. Okay. So for time to stand, for example, the leftmost group of bar graphs, you can see that in these participants, again, it was a group from ages 4 to 10. The average improvement was 2.9 seconds. If you move over to the 10-meter walk-run test, the average improvement was 1.1 seconds. And then in time to climb, it was 1.8 seconds. And that includes the mean improvement in these children, as well as a comparison to what would have been anticipated in an external natural history control. If you look over through, and the NSAA was even more impressive there, where comparing the two groups, there was a total of 4 points difference in the NSAA between the treated cohort and the natural history controls. This is all at dose level 1. Now we'll go ahead to dose level 2. And you'll see here that this was actually, again, only at the time of the data cut, a total follow-up of 9 months, because they were more recently enrolled, right? And smaller numbers having achieved each of those because of, again, more recent enrollment. But the data cut for all of this data for phase 1 and 2 was November 1st, 2024. And in dose level 2, only 2 participants had the 9 months of follow-up. But you can see when those two are compared on these time functional tests and the total NSAA score to their own external history controls, that there was a big difference. Okay, see here, and I can outline that for you. For the time to stand, there was an improvement of 2 seconds. Moving over in the 10-meter walk run, there was a 1 second improvement. In the time to climb the 4 stairs, a 2-second improvement in the treated group. And in the NSAA, there was a 7.3 increase in the NSAA total score in the treated compared to the controls. Yeah. So
Yeah, I'll take
hope,
over. I can take
okay,
over.
yeah. So I'll throw it back to Naz.
Yeah. Thank you. Thank you. Thank you so much, Dr. Kuntz. I appreciate it. And Dr. Kuntz can also, she's obviously a PI in our study, so we appreciate her kind of reviewing this with everyone. And obviously, we'll be open to questions at the end of this. What we wanted to talk about now is the phase 3 trial overview, because we are open and enrolling in this study. This is our pivotal study. We are enrolling boys who are over 1 year at screening. They have genetically confirmed Duchenne, and that's in mutations in exons 18 and above, but stay tuned. Very soon, things will be changing where we'll be able to enroll more patients outside of this exon bracket, and they must have no pre-existing antibodies. So if you have any questions about titers at this time, we're just positive or negative, but it's definitely on the horizon for us to try to attempt to see if we can kind of up that to include some kids with low titers. But at this time, it's either positive or negative. And again, there's some tweaks to the inclusion criteria for the children at the different age groups. So the only tweak that we really have here listed compared to what was for the phase 1/2 study was that for the 4 and over patients, the baseline NSAA should be greater than or equal to 16 total for the score. The endpoints for the pivotal study, so the primary endpoint is going to be the proportion of patients with greater than or equal to 10% of RGX 202 microdystrophin levels. So we're seeking to prove that all of our patients will get proper microdystrophin expression, recognizing that that microdystrophin will be easily quantified in the muscle tissue and expressed in the sarcolemma across the board. Our secondary endpoints are going to include the functional tests. So this is time to stand, time to run walk, 10 meters, and time to climb. We're prioritizing our velocities of that. So that includes velocity of time to stand, and also the absolute seconds. For the 1 to less than 4-year-old patients, we continue with the Peabody Developmental Motor Scale, but have also added the SV95C, the stride velocity 95th centile, as a wearable. And our exploratory endpoints will be the North Star, SV95C, and MRIs for the 4 and over patients. And the pivotal trial design is going to be pretty much identical, except for that we're extending it out to our endpoint for functional endpoint to be at 24 months after dosing. So we want to prove that first, that our microdystrophin expression is good, it's a good surrogate for clinical benefit, and that our clinical benefit is not just there, but it's also sustained at 24 months. And these are the locations where our study is up and running. So gold star for Dr. Kuntz right there in Illinois. And basically we have different sites in Virginia that are open up, Arkansas, Dallas, Texas, and California as listed here. And we plan to open up a few additional sites shortly. Next slide. So in summary about the Affinity Duchenne study, this pivotal study is a multi-center open label trial. There is no placebo control. We're using natural history controls for comparators. So it's an open label trial investigating RGX 202 as a potential treatment for Duchenne, and potential participants for the pivotal study can be referred to the Affinity Duchenne clinical trial sites on clinicaltrials.gov. And if you have any questions for us, you can email us at REGENXBio, and that is medinfo@regenxbio.com. And just wanted to finally talk about our Affinity Beyond study, because this is an interesting study that you might be interested in referring your patients to. This involves it's a non-interventional study. It's just checking for anti-AAV8 antibody assessments in boys with Duchenne. So this will enroll up to 200 US participants. We're going to be adjusting this protocol to allow for older patients, since our pivotal study is now in 1 and over patients. But at present, it's for 0 to 11-year-olds, and we're just checking for the presence of antibodies. And it is not discriminating against your mutation type. And if you are found that you are AAV negative, you could potentially use this result as a reference to entry, if it's within the correct time frame, into the Affinity Duchenne study for dosing. And with that, I'd like to say thank you so much, and go on to questions. And I see that we do have some questions in the chat. So Jessica, just let me know if you want me to get started or if you had any closing comments.
Please, if you could address the questions quickly, that would be amazing, and then we'll close. Thank you.
Okay, that's great. So I see some in the chat. So one question is, are eculizumab and sirolimus given even if the subject is antibody negative? Yes. So they are given prophylactically, not just, from a safety standpoint for sure. And I do think that this does play into our good safety profile. But also we theorize, obviously we can't totally prove this, but we theorize that it can also help with transduction of our vector. And here it says, do participants need insurance authorization to participate, or will there be no cost to participate? So there's no cost to participate. Even if you're not close to the site, we can cover travel. We have a budget to do that. Insurance authorization, it's good to have insurance just in case, God forbid, something happens, but obviously we would be willing to cover additional costs if there were a significant an adverse event that needed treatment during the study. And we'll just those two. Oh, that's it. Okay. So I think that's all I have. Are there any additional questions or comments?
Great. I think that we will leave it at that. Thank you so much. You shared wonderful information today. Thank you to Drs. Pacola, Dr. Kunst, and Dr. Dastigier for the insightful presentation for our medical professionals attending today's program. We hope you'll join us for future industry update webinars, and have a wonderful day. Thanks so much.